Welcome everyone to the fifth webinar of the Science of Aging Series 2021, a joint webinar series brought to you by Inside Scientific and the American Physiological Society. For this series, we've lined up a number of webinars, all focused on the science of aging being conducted by leading researchers around the world in their relevant fields. This is Sarah McFarlane from Inside Scientific, and I'm very pleased to be your host for today's event. Today's webinar is titled Quality, Quantity, and Timing, Regulating Healthspan and Lifespan with Diet, and will feature Dr. Dudley Lamming, an associate professor of medicine at the University of Wisconsin at Madison. In his presentation, Dudley will discuss his research involving how diets can affect metabolic health and longevity. Before we get started, we would just like to acknowledge our partners at the APS and the Alliance for Aging Research, and also to thank the sponsors of this webinar series for making it all possible. And without any further delay, I'm very pleased to welcome Dr. Dudley Lamming to the floor. Dudley, take it away whenever you're ready.
What should I eat? If I'm a mouse, my choices of what I should eat and when I should eat it are largely dictated in a lab environment by the scientists who are investigating us. And that makes the mouse an excellent model for studying nutrition and metabolism. Because studying in humans, of course, is much more complex. Humans are faced with what noted food author Michael Pollan referred to as the omnivore's dilemma, the fact that we could choose to eat a wide variety of different types of foods, varying in many different ways in terms of their quality. When asked what we should be eating, Michael Pollan said that we should eat food, not too much, and mostly plants. At least in the first part of that, the not too much, we've known that for a number of years, almost a century now, that restricting caloric intake is a very effective way of extending health and lifespan in numerous species. This ranges all the way from yeast up to rodents, as shown on the left. Looking at the lifespan curve all the way to the left, we see some ad libitum fed mice, whereas varying degrees of restriction extend the lifespan very significantly of those animals. Looking at the panel on the right, we see that there's changes induced by CR in terms of mortality, even in non-human primates in the Wisconsin study conducted by Rick Weindrich, Ricky Coleman, and Roz Anderson. So while we don't know that, that this extends the lifespan in humans, there's a lot of evidence that it certainly extends health span in all species, reducing incidence of many different age-related diseases, including cancer, cardiovascular disease, diabetes, and Alzheimer's disease, at least in some models. So today I'm going to talk about a few related stories. The first, we're going to talk about how calorie restriction works, and in particular the role of fasting. And then talk about a specific dietary macronutrient, protein, which is composed of, as building blocks, about 20 different dietary amino acids. And we're going to focus on the branched chain amino acids, leucine, isoleucine, and valine today. And at the end of each of those sessions, I'll give some conclusions about what our results may suggest in terms of a healthy diet. So an evolving thing in the research field has been the idea that when we eat during the day plays an important role in regulating health. And this has been based on work by Sachin Panda's lab as well as others, showing that restricting the portion of the day in which we eat is related to metabolic health. There's been several clinical trials now in humans showing that time-restricted eating, restricting the food-consuming portion of a day to 4 or 6 or 8 hours, has significant effects on metabolic health and can really improve metabolic health. Now, something that's interesting is that even though we have been studying calorie restriction for almost a century, it was only sort of realized fairly recently that calorie-restricted animals are also subject to time-restricted feeding. And this is just because in the laboratory environment, calorie-restricted animals are typically fed once per day, often in the morning, sometimes in the evening. And as a result, because they're hungry, they binge their food, and they can consume their food in about 2 to 3 hours. And thus, even though they're being fed once per day and we're not intending per se to fast them, we're collaterally imposing a very prolonged fasting period on these mice or even other animals. And a few years ago, work by Rafa DeCabo's lab in collaboration with Roz Anderson at Wisconsin and a number of others was trying to investigate how different diets that were fed to non-human primates had different effects on aging and survival. And as part of this study, what they found was that meal feeding the animals a meal that they would actually finish in the space of a day, had significant effects on lifespan. And so you could see here in all three of these curves, regardless of the diets, while calorie restriction extends lifespan very significantly, meal feeding, where calories are not really restricted, but the animals are fasted for roughly 11 to 12 hours per day, also had a positive effect on survival. And so this and other research suggested to us that maybe this collaterally imposed fasting that calorie-restricted animals are subjected to really has an important role in this effect. And so we wanted to ask if the benefits of a calorie-restricted diet arose solely from the restriction of calories or also from this imposed fasting period, or maybe a combination thereof. So a graduate student in my lab, Heidi Pax, sort of used a series of diets to investigate this possibility. And so she put mice on either an ad libitum fed diet, a diet that was a normal control diet, basically a normal facility chow, a diluted diet in which that chow is diluted 50% with cellulose. This had the effect of restricting calories by about 30%. Or two different regimes where calories were restricted by 30%, but either this was done in the classic way in once per day feeding, or we used an automated timer to drop meals at multiple times per day, eliminating the ability of the animal to binge all of its food at once. And what we found was that, as we expected, all of these animals gained less weight or lost weight relative to ad libitum fed mice when they were on these restricted or diluted diets. But you can see, just in terms of the patterns, that the effects of these diets on weight, lean mass, fat mass, and adiposity are not exactly identical. There's different things that are occurring. Now, we're really interested in how CR regulates glucose metabolism and insulin sensitivity. And so we perform glucose tolerance tests on these mice where the mice are all fasted for an equivalent length of time. And then we administer glucose and see what happens to their blood glucose level over the course of about 2 hours. And so what you could see is that as we expected, our calorie-restricted mice had improved glucose tolerance, a reduction in both fasting blood glucose levels as well as the total area under the curve, or AUC. And that restricting calories either via the diluted diet or via an automated timer where the binging period is eliminated had very similar effects. So calories alone seem to really mediate the effects that we see here on glucose tolerance. But interestingly, this wasn't the case with insulin sensitivity. When we administer insulin to the animals and we look and see what happens to blood glucose over time, a very widely conserved response to calorie restriction that's well documented in the literature is that calorie restriction improves insulin sensitivity. And you can see here in the black bar, the calorie restricted mice have a much, much greater dip in blood glucose when insulin is administered and a reduced area under the curve. But restricting calories in other ways, either with the diluted diet or the automated feeder, where there's not this prolonged fasting period, doesn't have the same effect. And so it seems like fasting is required for the improved effects of calorie restriction on insulin sensitivity. We're also interested in examining some of the other effects of a calorie-restricted diet. So one of the really interesting things that a calorie-restricted diet does is change how lipid metabolism and energy fuel source utilization occurs. So we were able to measure this using Columbus Instruments' CLAM system. This is a system that measures respiratory gases and in our setup also tracks activity and food consumption, and enables us in particular here to look at the respiratory exchange ratio, which is just a ratio of carbon dioxide and oxygen. It tells us something about what types of fuels are being used as an energy source. And so what happens here after feeding of traditionally calorie-restricted animals at the red arrow, what we see is that the RER increases dramatically in the calorie-restricted, that's the black, or the TR.CR, that's the automated feeder in red groups, whereas in the ad libitum fed group and the diluted diet group, it doesn't. And that's right in the center in the light phase. And so what that's indicative of is as the RER shoots above 1, what we see is that the calorie-restricted animals and these mice fed from a feeders as well have a burst of essentially lipogenesis. So carbohydrates are being oxidized very rapidly and used to form new lipids. And that's been explored in some depth by a number of groups. But our diluted diet fed mice, the mice that are not subject to this daily prolonged fasting or of any length, don't have that. And so it seems like that fasting period really reprograms how animals use the fuel sources that are available to them in the diet. And in the case of a prolonged fasting, they have a ton of lipogenesis and then use that energy during the rest of the day. But if they don't have that prolonged fasting period, that isn't what happens. They sort of burn a mixture of fuel sources continuously over a 24-hour period. We also can use respiratory gases to measure energy expenditure, and we can look at that either relative to a lean mass or whole body weight, and the results are perhaps not surprising, which is that animals that are fed an ad libitum diet use more energy. Calorie-restricted mice tend to be lighter and leaner and also, generally speaking, use less energy, and you could see that both in top over the 24-hour plot, basically all through the dark period of both before and after the feeding that's shown here. The animals with reduced caloric intake, regardless of fasting or not, use less energy. But although interestingly, you can sort of see that the blue group, the diluted diet fed animals, have sort of an intermediate pattern of energy use. They actually do use not quite as much energy as the ad libitum fed mice, but not quite as little as the CR fed animals either. And if you look closely at some of our body weight calculations, it seems like those animals might even be a little bit leaner on these calorie-restricted diets where there's not imposed fasting period. Now, Heidi then want to find out whether fasting alone was actually sufficient for a lot of the effects of a calorie-restricted diet. And interestingly enough, that is true. So she was able to train the mice to eat all of their food in about 3 hours. That's the red TR.AL group I'm going to show you here. And when we look at weight and body composition over the course of a few months, what we see is that this imposed fasting period where the mice eat only 3 hours a day and the food is then that they don't eat is then removed. They essentially have more or less normal caloric intake, but they gain less weight, they gain less adipose tissue, and they actually retain lean mass, which is a little bit different than the calorie-restricted alone animals. We see very similar effects of fasting and CR on glucose tolerance. Again, sort of recapitulating what we saw in the previous experiments with restriction of calories without fasting. But interestingly, fasting alone is capable of improving insulin sensitivity. And this is something that calorie restriction alone does, but you'll recall that the diluted diet and the diet fed by the automatic feeders did not. Here we use our metabolic chambers again. We look at respiratory energy, the respiratory quotient. And what we see is that fasting, again, right after you feed those animals, either for the fasting group or the CR group, you get a massive increase in RER, indicating that carbohydrates are being used almost exclusively as the fuel source and that active lipogenesis is occurring for about 6 or so hours. And then this animals subsist on those stored lipids during most of the day. And if we use those values to calculate fuel oxidation, we can see that overall a much greater proportion of the energy that's used by fasting mice, the TR.AL group, or CR-fed mice comes from fatty acid oxidation. And there's a lot more carbohydrate and protein oxidation being contributed to energy in the case of the Ad-Libitum-fed animals. We did a number of studies to look at the effects on molecular signaling. One of the most dramatic, though, is in terms of looking at transcriptional profiling. What we see is that there's very similar effects of both fasting and calorie restriction on the transcriptional profile of multiple tissues, including the inguinal white adipose tissue, or IWAT, and the liver. And in fact, fasting is not only recapitulates the molecular effects and can recapitulate some of these other metabolic benefits, but we then went on to conduct a lifespan study where we found that fasting is required for the beneficial effects of CR on both health span and lifespan. So if we take ad libitum, we took young black 6 mice starting about 6 months of age. We put them on either an ad libitum fed diet, the diluted diet that has no fasting period, or a traditional CR diet. And looking at how frailty accumulated in those animals over time, you can see that the calorie restricted animals are protected from an age-associated increase in frailty. And we also found that calorie-restricted animals are living longer, while animals fed this diluted diet, where calories alone are being restricted without fasting, are actually living shorter. And this is in accordance with a little bit of data in the literature as well. And so I think this really suggests that you really need this fasting period in order to get the benefits of a CR diet. So to conclude this portion, fasting and decreased energy expenditure really contribute to both the metabolic and molecular effects of a CR diet. Fasting alone is sufficient to see the effects of a CR diet on metabolism as well as on transcriptional profiles. And decreasing energy intake without imposing a fasting period during the day does not promote robustness, does not keep us from accumulating frailty, and it doesn't extend lifespan. So enough about calories. Calorie restriction really seems to be a very potent effect, and I think it's very important for us to understand how calorie restriction works as a tool. But in the society that we live in today, where there's an abundance of food at all times, it's very difficult to imagine that many people could really sustain a calorie restricted diet for very long. But when we think about a diet is actually made up of 3 major macronutrient categories: protein, fat, and carbohydrate. And there's been a lot of interest for many years about the idea that maybe there are portions of this diet that are healthier for us than others. In particular, about 70 to 100 years ago, there's a lot of focus on fats, and the idea was that fats, in particular types of fat, were really bad for cardiovascular disease. And more recently, as the amount of obesity and diabetes in the United States and around the world has really skyrocketed over the last couple of decades, there's been a focus on the idea that carbohydrates are bad for us because they drive obesity, they drive diabetes. And in all of this, protein has generally been suggested to be sort of beneficial, that dietary protein promotes satiety. And so there are fad diets based on the idea that if you eat large amounts of protein, it will decrease your caloric intake. And also, if you eat large amounts of protein, it displaces calories from fat and carbohydrates. And as mentioned, those have a bad reputation. But is this really true? Is dietary protein good for us? Well, an interesting study that really focused my attention on this was done by Samantha Solon-Binier and Steve Simpson and David Lacuter's labs in Sydney back in 2014, where they looked at many different groups of mice fed diets with different ratios of these different dietary macronutrients. What they found was that the mice fed low protein diets were actually the ones that had the longest lifespan. And if you look in the literature, well, there's a lot of very short-term studies suggesting that high amounts of dietary protein are good for insulin sensitivity, for building skeletal muscle, potentially in the elderly who might have sarcopenia. In the long term, looking in the general human population, low protein diets are really associated with improved health. Low-protein diets in an enhanced retrospective analysis were shown to be associated with decreased rates of cancer, overall mortality, and diabetes. In a very large prospective cohort study conducted in Europe over about a decade with 40,000 people, diabetes risk was shown to be about twice of that in the highest quartile of protein intake versus the lowest quartile protein intake. So there's definitely an emerging consensus that high protein diets may have some deleterious effects and low protein diets may be more beneficial, not only in rodents, but also in humans. This has also been shown to be linked in the case of cardiovascular disease and a number of other diseases. In large epidemiological studies have shown associations between high protein diets and cardiovascular disease. So to address whether this was really true in humans, that some of the metabolic effects of dietary protein could be seen in both species, we conducted a clinical trial with Luigi Fontana's laboratory at Washington University in St. Louis. And so Dr. Fontana placed people who were 53-year-olds on average, males, all with very low-grade prostate cancer, on either a control diet where they ate their normal diet at home, or they ate a protein-restricted diet where they were fed in the hospital cafeteria, and 7 to 9% of the calories came from dietary protein. And so we know exactly what they eat because they were fed in the hospital cafeteria. And so what was really interesting, what we found, was that there were really interesting effects on metabolic health. And so this study lasted for about 6 weeks. And the protein-restricted group lost about 2.5 kilograms, or a little over a pound a week. And by DEXA, we were able to determine that that was focused on fat mass. Blood glucose levels in these individuals also went down. Now, whenever we do one of these dietary studies, whether it be on calorie-restricted animals or PR animals or humans, we need to know what people are eating, because you could imagine that maybe the protein-restricted diet wasn't very palatable. Maybe they ate less. It turned out not to be the case. So in a agreement with the idea that protein promotes satiety, calorie consumption was actually significantly higher in the protein-restricted group. So we wanted to know if we could study the metabolic effects of protein restriction in a mouse. And so we designed 2 different diets where either 21 or 7% of calories came from protein. We were able to match these diets exactly for calories and fat, which we weren't able to do in the human study. This is a major advantage of doing work in rodents. And we can do lots of measurements in rodents, including looking at weight, body composition, glucose homeostasis, and putting these animals in metabolic chambers to look at activity and energy expenditure. And what we found was that mice fed a low-protein diet seemed similar in many ways to humans on our low-protein diet. So young mice fed a low-protein diet eat significantly more food, and as we look at their weight gain over time, these are young growing animals. And so in about 2 months, the mice on the 21% protein diet gained about 6 grams of weight, and the mice on the low protein 7% diet gained only about half that. We are also able to look at body composition. And what was really exciting was while both lean and fat mass were implicated, as you can see on the left, at 8 weeks, there was a very dramatic effect on fat mass. And so these animals had reduced adiposity overall, even though they gained both less fat mass and less lean mass. Using our metabolic chambers from Columbus Instruments, we looked at energy expenditure. As we anticipated, based on the fact that they're eating more food, more energy in, but weren't turning that into body weight, these animals are actually expending more energy. You can see there's a trend during both day and night, but that was statistically significant during the nighttime. We also looked at a number of different measurements of glucose homeostasis. These animals have a lower fasting blood glucose, just as the humans on a low-protein diet do. They had lower insulin levels to go along with that. And when we calculate HOMA2IR, which is essentially a measure of insulin sensitivity, the animals appeared to be more insulin sensitive. They had a lower HOMA2IR. When we conducted glucose tolerance tests, we found that the animals not only had lower fasting blood glucose, but a lower area under the curve during the test as well. in work that I'm not going to show you, we and others have basically believe that this is primarily mediated by improved hepatic insulin sensitivity and not a result of altered function of pancreatic beta cells, which is another important mediator of glucose tolerance effects. So we wanted to next drill down and try and understand what it was that really mediated these effects of dietary protein. And the approach that we took was to realize that dietary protein consists of amino acids. Amino acids are the building blocks of protein, and typically, while there's many different amino acids, we typically focus on the 20 common amino acids that are directly encoded by the nuclear genome and really form the building blocks of most proteins in the body. And so we used a series of synthetic amino acid defined diets to specifically examine the contribution of the essential dietary amino acids. My lab has now looked at all of those essential dietary amino acids, but the ones I'm going to talk about today are the ones we started with and are some of the most interesting. The branched chain amino acids, leucine, isoleucine, and valine, so called because of their structure. Now, why did we focus on the BCAAs? There's a couple of reasons, but one of the most important is that the BCAAs have been known to be associated with insulin resistance for a long time. So back in 1969, it was shown that BCAAs are elevated in the blood of people with type 2 or with insulin resistance. And subsequent work by many people, including Christopher Lynch, Chris Newgard, Susan Hudson, and many others, over the last several decades have indicated that this seems to be sort of a conserved thing, that BCAAs are typically elevated in insulin-resistant people and animals, as well as obese people and animals, and that BCAAs actually correlate with obesity and the development of type 2 diabetes. We also focused on the BCAAs for another reason. So we looked at amino acid levels in the people in our human protein-restricted trial. And interestingly enough, the levels of the BCAAs were significantly altered, lowering by about 5 to 10% in the blood of people on a protein-restricted diet. Whereas other amino acids were unchanged, and these included things like methionine, which has been proposed to be something that mediates not only the effects of PR, but also some of the health benefits of vegan diets. But we didn't see any changes in methionine. So how did we do this? Well, we made a series of 3 major diets that I'll focus mostly on today. A control diet, where 21% of the calories come from amino acids. It matches the 21% natural source diet that I talked about earlier, but is now made from individual amino acids. A low AA diet, where 7% of the calories are from amino acids. Again, the same amino acid profile as the natural source diet we used earlier. And a low BCAA diet, where we specifically reduced the levels of the BCAAs by 2/3 to match the level found in a low AA diet. Now, when I say that we made these from amino acids, I literally mean the individual amino acids were mixed together in a vat by our food vendor, Invigo. And the major advantage of doing these studies in rodents is that we can keep everything isocaloric, we can keep the same energy density, the calories and source from fats are constants, and we can, when we remove amino acids, like the BCAAs, we can balance them by adding either carbohydrates or non-essential amino acids. And we've done both, and we basically get the same results either way. So mice fed a low BCAA diet look very much like protein-restricted animals. They eat more. You can see here in the panel on the left that there's increased food consumption in both low AA and low BCAA fed animals. And they gain less weight. Over the course of 10 weeks, the animals here on the control diet gained about 6 grams, whereas the low AA and low BCAA diet fed animals gained much less weight. Again, we were able to look at body composition, and both low AA and low BCAA fed diets are associated with an overall reduction in adiposity. So you can see that the percentage of the body weight that's made up of fat shrinks, while the lean mass is more preserved. Restricting either all amino acids in the case of a low AA diet by 2/3, or just restricting the 3 BCAAs has very similar effects, beneficial effects on glucose tolerance. Here you can see those red and green lines overlap almost precisely, and the effect on the area under the curve is basically identical between low BCAA and low AA fed animals. Now, in some work that we actually just published a few weeks ago in Cell Metabolism and is available in the resource tab, we've actually now broken this down even further, looking at the individual branched amino acids to try and zoom in on which of the amino acids is really mediating these effects. And what we found is that, quite surprisingly to us, isoleucine is really the major effector of this, but there's also a role for valine as well. So if we restrict either leucine or valine by 2/3, you can see that there's a particular reduction in the case of the red low isoleucine fed diet. The orange low valine diet also has a trend downwards, and a low BCAA diet, of course, as we expected, also improves glucose tolerance. The thing that's really surprising here is that leucine restriction alone had no effect on glucose tolerance. And we actually tried higher levels of leucine restriction as well, for a variety of reasons. And actually, overall, there seemed to be no positive benefits from leucine restriction on glucose tolerance, even at a higher level of restriction. Dietary isoleucine in particular has a profound effect on body composition, and reducing dietary isoleucine or all through BCAAs really blunts fat mass accumulation as well as overall weight gain to a much greater extent even than valine and leucine restriction. You can see here had a trend towards actually an increase in fat mass. That's something that we've seen in one of our previous studies as well. Taking sort of a systematics view, looking at a heat map with many different parameters, including different measurements of body composition, different tests such as a glucose, insulin, or pyruvate tolerance test, what we could see is that isoleucine restriction or valine restriction largely seems to largely recapitulate the effects of a low BCAA diet, while a low leucine diet has the opposite effect, moving sort of a yellow direction in most cases, whereas the other diets move in sort of a blue negative direction. We've also done the converse experiment where we look at a low protein diet or a low AA diet in this case, and then we add back different branched amino acids. Here we've actually split out the graphs into two separate sets so that it's sort of easier to see, but this was all done as one experiment with 6 different groups. And what we found is that the control or low AA diet had the expected effects on glucose tolerance, with restriction of dietary protein really having quite profound positive effects on glucose tolerance. And we could blunt that by adding back either isoleucine or all 3 BCAAs. In fact, if all three BCAAs are added back, there's actually no effect overall on glucose tolerance, whereas we eliminate about half of the effect just through the readdition of isoleucine. In contrast, valine has a very small effect when we add it back, and as we would expect from our restriction regimen, you can see adding back leucine here has no effect on glucose tolerance in these animals at all. And so again, isoleucine and to a slightly lesser extent valine seem to be the key mediators of glucose metabolism in terms of the BCAAs. Looking at weight and fat mass, adding back either isoleucine or the BCAAs blunts very substantially the effects of a low protein diet on body weight and body composition. And again, valine and leucine add back do not. In fact, leucine add back actually made the animals weigh even less, which is quite an interesting effect overall. We don't truly understand yet why that is. Again, looking at sort of a systems view using a heat map approach, we could see that taking a low AA diet and adding back either isoleucine or the BCAAs makes a mouse very similar to a control AA fed mouse that's just eating a normal diet fed ad libitum, whereas valine readdition sort of has a mixed effect, and leucine restriction really doesn't recapitulate the effects of a control diet at all. In fact, in some ways, it actually has profound effects in the opposite direction. So we've been really interested in trying to understand what the physiological and molecular mechanisms behind the effects of dietary isoleucine on metabolic health are. And one of the things that we've investigated is the effects on glucose tolerance. So working with Joe Bauer's lab at UPenn, we performed hyperinsulinemic euglycemic clamp experiments to look at glucose production and glucose uptake into various tissues. One of the things that we found is that there's a very significant effect on hepatic insulin sensitivity. Under clamp conditions, you can see that low ILE fed mice have substantially greater suppression of hepatic glucose production by insulin than mice on a control diet. So to take sort of an unbiased approach, we conducted transcriptomics as well as metabolomics approaches, looking particularly at the liver. And one of the things that we noticed was that there was a very significant change, a very interesting gene, FGF21. So FGF21 is a hormone that's produced by multiple tissues, including the liver. And it's an insulin sensitizing hormone, as well as an energy balance hormone that's produced in response to fasting as well as some other stimuli. And in particular, one of the things that's quite interesting is that FGF21 has been proposed to be an endocrine signal of protein restriction. And FGF21 knockout mice have been shown to not respond to protein restriction in the same way as wild-type animals. So first, we looked at multiple tissues. We saw that there was an induction of FGF21 not only in the liver, but also in inguinal white adipose tissue and skeletal muscle. And we saw that there is a significant elevation in blood levels of FGF21 as well. So FGF21 is clearly being turned on by isoleucine restriction. Now, in addition to regulating hepatic insulin sensitivity, FGF21 also promotes energy balance. And one of the things it does is promote the browning and beiging of white adipose tissue. So here you can see inguinal white adipose tissue has significantly more multilocularity and smaller overall adipocytes than the inguinal white adipose of the low ILE fed mice. And look at gene expression, we see increased expression of multiple beiging and thermogenic markers, including UCP1. And we then used our Columbus Instruments metabolic chambers to measure energy expenditure in mice fed diets and where we were restricting either leucine, isoleucine, valine, or the low or all three BCAAs. You could see that both during the light cycle and the dark cycle, isoleucine restriction, valine restriction had a significant effect, really boosting energy expenditure. And so overall, the idea here seems to be fairly convincing that FGF21 mediated events may be occurring and may be responsible for some of the effects that we see in low isoleucine fed animals. So to really test this, we used mice where we knocked out FGF21, and we looked at multiple phenotypes. So one of the things that's really interesting is that a low isoleucine restricted diet, just as a low protein diet, induces an increase in food consumption. You could see that over on the left in the wild type animals. But in the FGF21 fed animals, that is completely blocked. And you can see that quite clearly. Now, we also wanted to look at energy balance. And so we did that again using our metabolic chambers. And what we found was that in the wild type animals, the low isoleucine fed diet, as we expected, and when we looked at it in our normal survey of the multiple diets, we saw a very significant increase in comparing the white bar to the red bar in both the light and the dark cycle in terms of energy expenditure. But this was blunted or blocked in FGF21 knockout mice. And so you could see that there's a statistically significant difference between the wild type animals on a low isoleucine diet and the FGF21 knockout fed animals on a low isoleucine diet in both the light and the dark cycles. Overall, it looks like FGF21 accounts for about half or maybe even a little more of this increased energy expenditure that's mediated by a low isoleucine fed diet. So FGF21 definitely seems to play a role in this response. It doesn't affect everything, though. So looking at glucose tolerance, we found that glucose tolerance, the improvement in glucose tolerance on a low isoleucine diet was not mediated by FGF21. So the wild-type animals as well as the knockout animals responded equivalently with improved glucose tolerance on a low isoleucine diet. And so there's still more to discover about how a low isoleucine diet promotes metabolic health and what processes mediate this. So in conclusion, and in that part, I think that overall, I've told you that dietary protein and restriction of dietary protein promotes metabolic health in both humans and rodents. And in our rodent work, it seems like BCAA restriction or restriction of just isoleucine can really recapitulate these effects, potentially even being more powerful in the case of isoleucine restriction. So the next question that we wanted to focus on is, can we promote health and longevity by reducing consumption of branched chain amino acids? And this is some work that we published a couple of years ago in part, but to sort of give you the really cool take-home experiment, what we were able to do is we were able to take wild-type black-6 mice and fatten them up, make them quite obese by feeding them a Western diet. It's a high-fat, high-sucrose diet that has some added cholesterol as well. And these animals become much more obese than chow-fed control animals. And then we randomized these Western diet-fed animals onto a series of amino acid-defined diets where we mediated and changed either the levels of branched amino acids alone, or we examined protein restriction. And what we found was actually quite interesting. So in this trial, one of our major readouts was going to be weight as well as glucose tolerance and insulin sensitivity. So here, just looking at the weight, you can see that Western diet fed mice, they're switched to either a Western diet control amino acid defined diet or Western high BCA diet, stay fat and continue to gain some additional weight during the time that they're being fed this high fat diet. Whereas the mice on the control amino acid defined diet stay essentially lean. But if we restrict just the branched amino acids or just dietary protein by 2/3, we get a very dramatic decrease in weight. And so this is not associated with big changes in food intake. In fact, food intake actually increases as we go out further on these diets. But the animals are instead seem to lose quite a bit of weight, even though they're eating plenty of food. And even though overall they seem to be in good balance in terms of the amount of food that they're eating. So what's actually mediating this effect? Well, we see increased energy expenditure when we place them in metabolic chambers. And when we look at the individual diets, restricting just leucine, isoleucine, or valine, similar to what we saw in our lean animals, we see that leucine doesn't really play a role in this, but valine and isoleucine do. And so just restricting either isoleucine or just restricting valine dramatically decreases weight and fat mass, equivalently essentially to the effect that we observe on a low BCAA diet, maybe even more so in the case of isoleucine restriction. And so it seems like, although we still don't understand the physiological processes exactly, that there's a very dramatic effect of isoleucine. Some of this may go through FGF21. And of course, an important question is whether or not this will work in humans. Since we started working on this, there's been a couple of very short-term trials done of BCAA restriction in humans in Europe that definitely suggest that maybe in type 2 diabetics, there may be some positive effects on insulin sensitivity. We've been really interested in taking a more broad view here, which is where we've been enrolling people in a diet study where we restricted BCAA intake by about 50%, and then examined the effects on lots of different metabolic parameters. And we originally were focusing on people with prediabetes as a good population to examine. And so we're still wrapping up this study. Overall, it seems like this is a feasible intervention, and we're hoping to expand it in the future to include enough subjects to get good power to measure metabolic outcomes. But in the meantime, we have done some epidemiological work with collaborators at SHOW, the Survey of Health of Wisconsin, and its director, Dr. Kristen Malecki. And what we found there is that there's association between dietary isoleucine levels and body mass index, BMI, in people who live in Wisconsin. So this is almost 800 people who we have electronic health record data for, as well as data on what they tend to eat. And what we see is that a relatively small change in the isoleucine levels of total protein. So for instance, from 4% to 5%. Of the total protein, which is a 25% increase, is associated with a beta of 2.5. So BMI would be expected to increase by about 2.5 in those individuals as they increased isoleucine consumption. People tend to really want to be interested in what types of foods these are. We're still working on assembling some different dietary plans, but isoleucine tends to be lower in turkey and emu, as well as certain plants and nuts, and higher in food that is traditionally thought of as bad, like fast food and brats. And so definitely, though, we're interested in trying to design some more achievable diet plans that can focus on low isoleucine consumption. Finally, I want to tell you about our lifespan studies. So we've been really interested as to the effects of these types of diets on healthy aging. And what we found is that if you start young in life and you switch mice to either low protein or low BCAA diets, we have really profound effects on the lifespans of males. So we see about a 30% increase in median lifespan in male animals fed a low BCAA or low A diet from early in life, while in females, we don't see this happening. Later in life, intervening in more midlife for a mouse, so probably somewhere around 40 to 50 years of age for a human would be the equivalent. We don't see big effects on survival, but we do see that there's some anti-cancer effects of a low BCAA diet, as well as continued improvements in metabolic health. And when we look at frailty in these animals, we actually see that males in particular, started at a young age, are protected from age-associated increases in frailty when they're fed a low BCAA diet. And for that reason, we've looked particularly at the skeletal muscle of low BCAA-fed mice. And interestingly, we see sexually dimorphic effects on gene expression and molecular signaling in these animals as well. So there's about 1,000 genes changed in male skeletal muscle on a low BCAA diet, but only about half of that in females. And what genes are actually changing is actually quite different. So here's a principal component analysis showing that while control mice, males and females, tend to sort of overlap, you could see that the low BCAA diet sort of pulls them in different directions. And we could see this if we do some pathway analysis. You could see that both males and females have a lot of different pathways that are changed, but the exact pathways that are changed don't tend to overlap. And so there's definitely quite a few changes there. Now, my own expertise back before we started working on diets was really on the Mtor signaling pathway, which is a protein kinase that's very well known that when you decrease it, it extends lifespan in diverse species, including mice. And Mtor signaling, interestingly enough, was predicted to be altered in the male mice. We actually saw an upregulation of Mtor signaling, but when we looked at the individual genes that were being changed, what we observed was that Mtor genes that inhibit Mtor pathway signaling were the ones that were being upregulated. And so when we look in the skeletal muscle at the phosphorylation of Mtor substrates, we actually see that low BCAA-fed males have decreases in the phosphorylation of substrates, including S6 and S6K1, and overall a decrease in Mtor signaling in multiple tissues. And that turns out not to be true for females. So in females, there is essentially no change in Mtor complex 1 signaling. And so that correlates very well with the lifespan phenotypes that we have. So overall, in conclusion, decreasing dietary protein or BCAAs or isoleucine alone seems to increase metabolic health. Restricting BCAAs or restricting isoleucine or valine in the context of diet-induced obese mice really rapidly restores metabolic health and normalizes weight. And we're really interested in trying to figure out if that's true in humans as well. And we found that consumption of a low BCAA diet promotes the health and longevity of several progeroid mouse strains, which I didn't show you, as well as wild-type mice that we did look at. So overall, we think that people may want to rethink what they eat and potentially focus on consuming lower BCA-fed foods. If, of course, humans respond to dietary protein BCAAs the same way that mice do. And we may also want to rethink when we eat. A lot of people who have done calorie-restricted diets or diet plans don't incorporate fasting. They spread their meals out during the day. And so if humans respond to fasting in a way similar to the mice, that fasting component may be very important to include. So I'd like to thank everyone involved in these projects, as well as the organizers here for giving me the opportunity to tell you about our work, and the people at Columbus Instruments for helping us with our metabolic chamber system for many years now. This work was primarily led by Nicole Richardson, a former graduate student who's now at the University of Washington, and Charles Yu, who worked on the Isoleucine restricted project, now a postdoc in Perry Prugsever's lab at Harvard. And Heidi Pack, a graduate student in the lab, did all of the calorie-restricted work that I talked to you about at the beginning of the talk. And I'd like to thank all our funders, particularly the National Institute on Aging, the VA hospital, and all of our great collaborators as well. And now I'm ready to take any questions.
Awesome. Thanks so much for that, Dudley. And we are really excited to kick off our Q&A here. We have a lot of fantastic questions from the audience, and so we would like to keep those coming. Okay, so Dudley, are you ready to answer your first question?
Yes, I am.
Great. Okay, so someone has asked, what time of the day did you conduct the GTT and ITT?
We actually conducted the GTTs and ITTs for our branched amino acid and protein restriction experiments, primarily in the morning after an overnight fast. For our calorie restricted experiments, we tried a couple different ways to sync up the feeding parameters. Since all the mice are at different regimens, it's a little bit tricky to do that. Usually we did it after an overnight fast, but sometimes we also did it after refeeding so that the mice all ate at approximately the same time. It doesn't really matter, as it turns out, whether you do it for the calorie restriction experiments in the morning or later in the day. You get pretty much the same set of results.
Okay, great. We've got another question here regarding fuel oxidation. How can you stimulate amino acid oxidation?
That's an interesting question. So branched amino acid oxidation can be stimulated via a drug called BT2 that essentially inhibits an inhibitory kinase. So by inhibiting that inhibitor, you stimulate BCAA catabolism. All of the various different amino acids, for the most part, have different catabolic pathways. And so you would, for each type of amino acid, you might need one or might need to modulate one or a number of different pathways. I don't think there's a general way of stimulating amino acid or protein catabolism, but there could be. I'm just not aware of it.
Okay, great. We've got another question here. We'll comment and question. I was surprised to see that the calorie-restricted group, which was fed once per day, didn't enter torpor. Do you know why that is?
Yeah, so mice typically don't enter torpor, which is essentially a state where they're sort of huddling in the corner and shivering when they're calorie restricted. And so they sort of adapt, and we step down the calorie restriction by about 10% per week to give them that time to adapt. So they don't really have a torpor-like response. Now, longer periods of time without food, of course, you will eventually enter torpor. But calorie restriction, as is traditionally done with once a day feeding, and sort of giving the mice a chance to adjust, they don't do that.
Okay, great. The next question here, does classic calorie restriction improve cognition in the aging brain, and can calorie restriction reduce the incidence of Alzheimer's disease?
Oh, that's a very interesting question. So certainly there's some evidence that calorie restriction in mouse models of Alzheimer's disease, and there are many different models of the disease, um, does have some positive effects with respect to the development of plaques and tangles and loss of cognition in mouse models of AD. And I believe there's some evidence now that in rhesus macaques, there's some inhibition of normal Alzheimer's / dementia as well. So one thing that we did do, and I think I didn't show in our calorie restriction experiments, we did some cognitive testing on the mice. And we found that indeed we saw preservation of cognition and improved learning behavior in mice that were fed once a day on a CR diet, and not mice fed the diluted diet where calories are restricted, but they don't have that fasting period. So that's a great question.
Awesome. We've got another one here. This question is, have you looked at markers of oxidative stress in calorie restriction or fasted mice? And if so, what role do changes in reactive oxygen species have on glucose tolerance or insulin sensitivity?
So that's a really interesting one too. So the first thing to point out is that there's definitely an evolving recognition in the aging field that the ROS theory of aging, that reactive oxygen species accelerate aging and DNA damage and the hallmarks of aging, is definitely incomplete. And so Michael Ristow's work on mitohormesis has shown that calorie restriction does in fact induce a little bit of oxidative stress in multiple species. And at least in worms, that oxidation stress is required for the effects of CR on lifespan. So the model there is that by inducing this low level of oxidative stress, you induce sort of a hormetic protective response against many different types of stresses. Now, certainly, there, mouse models that have high levels of oxidative stress typically have shorter lifespans or develop various diseases. So we haven't looked in this particular set of mice, but I would expect that both in calorie-restricted mice and fasting mice, there might actually be a little bit of an induction of oxidative stress, or at least an induction of oxidative stress response genes that help detoxify. But we haven't really looked at that, and that's something that we could definitely look into in the future.
Yeah, that's great. All right. There is another question here. With a low protein diet, were there any changes noted in the autophagy in muscle, islets, or hepatocytes?
So we didn't notice changes in autophagy, but in our mice where we did our lifespan studies and did transcriptional profiling in the muscle, we definitely observed decreased mTOR activity in the muscle as well as adipose tissue. And you would normally expect that would be associated with increased autophagy. And so we saw changes in ULK phosphorylation, but we didn't look at autophagy markers per se. But you would expect that there might be more autophagy. And typically speaking, quote unquote, younger mice actually have more autophagy anyway. There's sort of an age-dependent decline in autophagy. And we'd expect that the anti-aging interventions, if they extend lifespan, probably delay that as well. But we haven't looked, and that would be something really interesting to look into in the future.
Okay, great. The next question here is, have you tested the influence of temperature on the metabolic effects of isoleucine?
Oh, yes. So that's an interesting one. So one of the questions that we got a lot from people when we started looking into isoleucine restriction was whether or not this would still work at thermoneutrality. So mice that are housed at room temperature essentially are not at thermoneutral. They need to expend some amount of energy to keep their bodies warm. And that's similar to humans too. For the most part, humans don't live in a thermoneutral environment. We live in an environment where we have to expend at least some energy or add clothing and so on in order to regulate our body temperature correctly. So we place the mice at thermoneutrality, and basically, although all mice at thermoneutrality, including ours, eat less because they don't have to spend as much energy keeping each other warm, all the metabolic effects, the improved glucose tolerance, the improved insulin sensitivity, the changes in energy expenditure, as far as we could tell, all of those were still preserved at thermoneutrality. So this seems not to be an effect that's modulated by body temperature for the most part.
Okay, great. We still have tons of questions, so we're going to go a couple minutes over. But if you do have to leave right on time, which is right about now, we do invite you to please participate in that survey before you go. Okay, so we're going to answer a couple more questions here. This question is actually directly for you, Dudley. How has this research changed your own eating patterns?
Oh, that's an interesting one. So definitely I've sort of been more interested in thinking about the isoleucine content or content of amino acids in general and food. It hasn't changed my eating habits that much. As a vegetarian, I already consume probably a little bit less protein than the average person.
Awesome. Okay. I thought that was just such a cool question. There's a little bit more sciencey questions here. This question here is, how is the effect of isoleucine on decreased fat in body composition? How is it mediated?
That's an interesting question. We think that FGF21, which is this key energy balance hormone, definitely has some role in the mechanisms of this effect. But there's definitely a large number of effects that are FGF21 independent. And things that we are looking into is trying to understand that. And so, there's hundreds of genes that change in the liver when we restrict isoleucine. Many genes, almost 1,000 or so of a change in skeletal muscle when we restrict branched amino acids. And so there's lots of different pathways that are being engaged, and we're trying to figure out what the role of Mtor may be, what the role of FGF21 may be, and what other new pathways might be being engaged. So that's something we're really expect to be looking into pretty heavily over the next few years.
Great. Another question here. Why did you decide to feed the mice at the beginning of the light phase, since they normally eat during the dark phase?
There's sort of two reasons for that. So one is feeding the mice at the beginning of life stage is very well known to work in terms of extending lifespan and is widely utilized. So the NIA's aging colony that makes aged calorie-restricted mice available to investigators is feeds at the beginning of a life cycle, and that's sort of a standard technique there. But more broadly, when we want to think about how to line up the different feeding regimens, so mice that are fed ad libitum essentially eat their last meal right before the lights turn on. And so if we feed the CR mice right when the lights turn on and then wait a few hours, all of those mice, the ones that have eaten ad libitum overnight, as well as the CR-fed mice, are essentially synchronized in terms of their feeding pattern. And so that gives us another advantage to that technique.
Okay, fantastic. And just looking at the time here, we don't want to go too far over, so this is going to be our last question. This question is, how would endurance exercise for glycogen depletion affect the fasting versus ad libitum feeding models?
Well, I'll just say overall, there's a lot about the interaction of our studies with exercise that we don't understand yet. So, dietary protein overall is pretty heavily consumed by athletes and without any particularly obvious detrimental effects. And so for the most part, we think that exercise is probably going to be protective against the effects of excess dietary protein, maybe against isoleucine as well. Trying to figure out exactly how to incorporate those types of exercise studies into our diet paradigms in a thoughtful way is something that we've been thinking about for a while. And I think the same thing could be true with calorie restriction. And I think it's important because most of the, very few people overall do CR, but most of the people who are on calorie restricted diets are also very interested in exercise. So that doesn't directly address glycogen, but I think overall this entire interaction between diet and exercise is something that needs a lot more study. And I think it's quite interesting.
Yeah, that's fantastic. Well, I just wanted to thank you again for all of your insights today. It was really a pleasure to have you with us.
Thank you so much for having me, and thanks Chris at Columbus Instruments as well for helping to recruit me for this.
Great. And I also wanted to thank everyone for joining today, taking time out of your busy schedules to attend the webinar. Again, as I mentioned earlier, the slides and a recording of today's webinar will be made available soon, so look out for an email regarding those resources in the near future. And finally, if you haven't already, we invite you to please take a moment to provide your feedback on the survey and let us know what webinar topics you'd like to see in the future. In closing, thank you again for taking part in this Inside Scientific webinar produced in partnership with the American Physiological Society and sponsored by Columbus Instruments. And we look forward to having you with us again soon.